evening, everyone. I hope you have all a great afternoon. Can you hear me? No? Maybe I'll just take it. Is it better? Okay. So hopefully we'll all fit in. I think this is the first time the house is completely full and people have to stand. That's a good sign. So without any further ado, I would like to welcome you here. It's a great pleasure to have another MENDEL lecture of the, I think this time it's a 12th series of the MENDEL lectures in 2015-16. It's the second lecture of this year. And it's a great pleasure to introduce Aaron Ciechanover. He was born in Israel. He did his bachelor and master in Hebrew University in Jerusalem. Then he served his military duty for 3 years and then continued his PhD in Technion. It was in biochemistry, if I'm right, where he was in the lab with Hershko and Rose, discovering the conjugation of ubiquitin to protein as a target to target proteins as a signal for protein degradation. They discovered the molecular mechanism of and proposed the model of this conjugation for possible degradation and regulation of protein stabilities. He then continued with a postdoc in MIT in his further characterization of ubiquitin, which during the years was shown as essential for really protein stability and also paradigm for many of the mechanistics of pathologies of diseases like cancer or neurological diseases. But not only that, it also seems to play a very important role in regulation of various kinds. As an example, I tried to write down some of the awards that he was honorated. So in 2000, he received Albert Lasker Award, followed in 2003 by Israel Prize, and in 2004, which you probably more know, is a Nobel Prize in Chemistry, together with Avram Hershko and Arvin Rose. He's an honorary member of many of the academies, so just name few. It's Israeli National Academy of Sciences, also the National Academy of USA, National Academy of Sciences, American Academy of Arts and Science, and also Russian and Chinese Academy of Sciences. So I think that's pretty a good list, what his achievement, and I'm really looking forward to Aaron's presentation. So the audience is yours.
It's okay? Can you hear me? Okay, good afternoon. What I shall try to do in the next hour or so is to tell you the story of the discovery of the ubiquitin system. But since it's really, the system is exploding these days, there are more than 250,000 papers on the system these days. I will condense it, will not go into any details, but do divide the lecture into two. In the first part, I'll tell you how at all we thought that there is something hidden behind. What is it that people walk into the lab and feel that something is missing? Not that it's big, not that it's important, but that there is a problem that it still needs to be solved. And in the second part, since I'm a physician, I will jump very quickly to our contemporary, to current days, and tell you how the system was converted into a major drug development platform. And there are many drugs in the market already, but the future will be much richer than that, because I think that in recent years, or in the recent months, scientists have broken the ceiling that prevented them from developing drugs to target the ubiquitin system, and it looks like we are going to explode also on drug development in that sense. So I'll try to do the two things. I will not go to tell you what we are currently doing in the lab. I think these are details. You can read papers. I mean, this is less important. So, controlling the electronics. Okay, so what is it? Okay, so this is our city. We are about 3.5 hours from here by flight. Tomorrow I'll be there. And it's the city of Haifa. It's the very east side of the Mediterranean, and it's a beautiful city. This is the Baha'i center. So the Baha'i that were persecuted in Iran smuggled the body of their founder, Baha'u'llah, and at the turn of the century buried him here. And then they built a beautiful golden dome and beautiful garden that became a UNESCO heritage site, and it's really gorgeous. But the city itself is really beautiful. So, destruction. I mean, people were, in science you need also to be a strategist. Everybody, during the period that we entered the field, was busy with construction. It was the central dogma of biology, how DNA is transcribed into RNA and how RNA is translated into proteins, and everybody was mesmerized by the genetic code. And nobody cared about destruction, so we decided to go to the other side, and I'll tell you about the milestone, what attracted us to this side. But if we look today at the map of destruction, it's really huge. And I call it the destruction for the sake of construction, because there are three major destructive systems in the cell that are all awfully important. One of them is obviously the apoptotic system, which is destroying the entire cell, but only when damage occurs or when we need it for development. For example, in order to remove the interdigital tissue that connects it during the embryonic stage, because if this tissue will not be removed, we shall have fused fingers. It's called syndactyly. So we know that apoptosis is important for development. Apoptosis is important for removal of damaged cells, of malignant cells, and so on and so forth. So we all know that apoptosis is awfully important. Then we have the lysosomal system that is also critically important, and it's important for degradation of two types of proteins. One is extracellular proteins that are coming from the outside. They can come by pinocytosis or phagocytosis, or by receptor-mediated endocytosis. And they're very important for signaling, for example. So insulin, or the EGF receptor, binds to a receptor, signals to the cell, and then along with the receptor, it's going into the cell, into endosomes. From the endosomes, it's going to the lysosome and degraded. This is a mechanism of signaling, of set off the signaling. So in order to turn off the signal, you need to remove the whole complex, and removing it away from the signaling mechanism. So it's an awfully important also to bring cargo into the cell, to bring the iron on the transferrin receptor, to bring cholesterol on the LDL receptor. So all these mechanisms of absorbing extracellular protein for either signaling was a building block is mediated by the lysosome. But the lysosome recently has become even more important as the mechanism of autophagy has been unraveled, and there are several types of autophagy. There is microautophagy, where there is the lysosome pinches off droplets of the cytosol, convert them into vacuoles, and then the vacuoles open up and pour their contents into the lysosome. But there is also stress-induced autophagy, which is called macroautophagy. When we stress the cell, we starve it, or we expose it to lower oxygen tension, and so on. We start to see double membrane forms, and the double membrane engulf the cargo. And the cargo can be specific. It can be ER. It can be mitochondria, for example. So there is a specific mechanism for mitochondrial removal from the cell, from faulty mitochondria, which is called mitophagy. We see it in Parkinson's disease. I will not go into it. But autophagy has become extremely important. And then obviously the ubiquitin system that I'm going to talk about. And the ubiquitin system talks extensively to the two systems, because elements of the ubiquitin system, mostly ubiquitin ligases, and ubiquitination occurs both for stimulating the apoptotic mechanism and also for stimulating autophagy. Actually, parallels of the ubiquitin systems are sitting in each of them. So it's hard for you to understand what are parallels, but there are ubiquitin and ubiquitin-like system and so on. So the system kind of outreached to the two destructive processes. So now, as you can see, destruction has become awfully important as a defense mechanism, not only against damaged cells, but also to provide nutrients to the cell, removing proteins that are not necessary anymore, and stimulating other processes. So that's compared to 30 years ago, when people thought, well, destruction, let's leave it to the grade C scientist. Who cares about destruction? Because we are really the geneticists. Here I say it in the birthplace of modern genetics, but people really didn't care, and we thought that this is nevertheless important and started to dig into it. So why at all to degrade proteins? Why we need to destroy proteins? Why not to take, we grow up, we are being born, and then we grow up at the age of 15, 16, 17, we attain our final weight, and then we are living hopefully for 80, 90 years. Let's take all the proteins and live with them. Well, we cannot do it because proteins are extremely sensitive, as you all know. They are sensitive to temperature. We are living in awfully high temperature, 37 degrees. This is intolerable for proteins. Why we are living in 37? Well, for proteins, it's better to live in minus 18. But we cannot live in minus 18, not because it's cold, but because biochemical reactions will not be running at minus 18. So 37 is an optimal temperature. We are living in 21% oxygen. Well, we cannot live in less than that, but oxygen is deleterious and toxic to proteins. It oxidizes cysteine residues and methionine residues, and so all SH groups are sensitive to it. But so there is, as you see, we are paying a price for life. So part of the price is the fact that proteins are damaged all the time. Another part that we have to keep the temperature above the body temperature, above the environmental temperature, so we have to generate heat all the time. So there are many, if you think about it, life is not for free. I mean, we need to pay in many different coins in order to survive, and trashing and exchanging and renewing proteins is a heavy price. Now, it's extremely heavy because daily we are destroying 6 to 8% of our proteins, 6 to 8%. Now, some of the proteins are very long-lived, like hemoglobin. It lives for 120 days. In the erythrocyte, actin and myosin can live 48 hours, depends on the cells. In muscle cells, they live more. But some proteins are changing for 10 times an hour, like transcription factor. It depends on the importance of the proteins in regulation. Take P53, for example, or other transcription factor, they change several times an hour. So we typically, people who are working on proteins monitor half-lifetime. What is half-lifetime? The time they take to 100 molecules to become 50. Well, they never become 50 because 50, once they destroy, new ones are coming. So there is a steady state. Typically, there is a steady state. But nevertheless, how long it takes to 100 proteins to remove, to the cell, to remove 50 of them. So for some proteins, it's 1 minute. So in 1 minute, it's half-lifetime. And for some proteins, as I said, it's days or year or months. But on the average, we are exchanging 6 to 8% of our proteins a day, which is an amazing number if you think about it, because it has many implications. Because what is today, 22nd of October? So if a month ago, the 22nd of September, I was composed basically of completely different proteins. So I'm new, young. Well, you too, not me. So what does it mean to renew our proteins? I mean, there are several questions. First of all, aging. Why do I age nevertheless? So this is one issue. If I'm new, so how can I be old? Then another question is how we can change the hardware without changing the software. Think about it. People don't think about it. How is it that I still remember what was 2 months ago or 3 months ago? How the higher function of the brains, like intelligence, emotion, memory, whatever, feelings, are being kept despite the fact that the hardware is completely different. Completely different. We are changing our proteome rather frequently. So there are many theories about it. It's too complex to approach. People think about the Stanley-Prussian era theory, about imprinting, like prion diseases, that proteins can imprint knowledge, so-called, or information to one another, but we don't know. But the matter of the fact is that as we speak, we are exchanging our proteins. So we are destroying and renewing them all the time. So one thing is quality control. This is a good reason. Then control of processes. Once proteins are not needed anymore, we trash them. So these are healthy proteins, but we don't need them because otherwise there will be a disturbing process. I think about cell cycle. Cyclins or cyclin-dependent kinases or cyclin-dependent kinase inhibitors. They drive the cell cycle, the cell enter mitosis, or moving from G to S, from G1 to S to G2 and then to M. Each step has its own proteins. Once you move, you remove the proteins that drove you into this cycle. And then once you are entering G0, they have to be removed. And once the cell exits G0 and enters again cell cycle, so those proteins are healthy, but we don't need them. And actually, if we keep them, I will totally confuse the cell or stop it or accelerate it. So we need to remove not only faulty proteins, but also healthy proteins that are sitting on the way of the process. And obviously, to maintain the differentiated and morphological state of tissue, we need to remove proteins that are from the embryonic state that are not needed. So the liver doesn't need eye proteins and vice versa. And those proteins are coming back during malignant transformation. So in teratoma, for example, in malignant teratoma, we see teeth, muscle, bones, and so on. So this is kind of a tumor that really de-differentiate, brings back the information. So the founding father of the field in many ways was Rudolf Schönheimer, a Jewish scientist that lived in Germany. And like many scientists, was fired and then kicked out of the country and went to the United States to work in Columbia University. There are many manuscripts written on the period. This was kind of the gift that Hitler gave to the West. And many scientists actually that boosted American science at that time came from Europe. Europe was the birthplace of modern science, but not for too long. Unfortunately, Albert Einstein was one of them, and many books and monographs were written about this period, about the emigration of Jews from Europe. And he committed suicide because of some, but he is the one that really discovered protein dynamics. By simple experiments, he gave labeled amino acids to rats and found that the amino acids are incorporated into proteins and then phasing out. So now for us it's like, wow, it's easy. Everybody does pulse chase experiments in the lab, but for him it was new, and for the scientific community it was new. People thought until the time that proteins are stable, as I told you at the very beginning, why to change proteins, and the whole idea of dynamics was new at that time, and he concluded that not only proteins, but also other macromolecules like sugar and lipids are in a dynamic state. He issued a small book that is called The Dynamic State of Body Constituents, and then there was some issue in the family. He committed suicide, and everything stopped for a long while. And we are jumping to the mid-50s. In the mid-50s, Mel Simpson at Yale discovered that protein degradation requires energy. So this is an experiment where proteins are degraded. Doesn't matter, just monitoring. And once he changed the atmosphere in the incubator from oxygen to nitrogen, he went from A to D, protein degradation ceased altogether, which was very strange thermodynamically. Remember, we are using proteins as a source of energy, right? We are eating good meat or whatever, dairy products, and we are decomposing it in the gastrointestinal tract to amino acids, and we derive the energy. So why to put energy in order to degrade high molecular macromolecules to low molecular, to how high energy macromolecules to low energy small molecular weight ingredients like amino acid? It didn't make any thermodynamic sense. And it is true, even in the cell, when we are degrading proteins, energy is derived, but the process itself requires nevertheless initial investment of energy. Think about driving a car downhill. Actually, you don't need any energy. If you turn the switch off and you lift your leg off the brake, the car will go down, no problem, but it will be a short trip. In order to control the speed and to avoid crashing and whatever, you do need to invest energy, almost as high or as the amount of energy required to take the car uphill or the same hill. Why? Because we need to control. So think about control, cell, in order to control processes. Otherwise, the cell will be destroyed. If the cell will start to proteolyze itself uncontrolled, the cell will die. Now remember, we are talking about faulty proteins. Remember why we degrade proteins. So we need to be selective. So already, when we think about the function of protein degradation, the word selectivity and specificity is embedded in it. Let's say that in the cell there are 1,000 molecules of hexokinase, any enzyme, doesn't matter. What did people told me today on RNA polymerase II? Who told me that? I don't remember. Doesn't matter. So let's say that RNA polymerase II, there are 1 million molecules in the cell, and one becomes denatured. We need to pull out this single molecule and degrade it and leave the 999,999 molecules alive and pull out only one. How the cell does it? If there will be just trypsin or chymotrypsin floating around, the cell will be destroyed. So by specificity, we need to invest energy. So the cell doesn't obey, well, it obeys the laws of thermodynamics, but nevertheless, in order to achieve the laws of thermodynamics are what we call free-floating laws. But nature is not there. Nature added control layers on top of it. So the laws are being kept. But nevertheless, they are kind of circumventing routes that need to be exercised. So this is one property that caught our attention when we started at the mid-70s. Energy. Where is energy? So we knew that it must be different than the process that occurs in the stomach, because in the stomach you can put everything you want, it will be degraded. But in the cell, this is not the case. Then came Christian de Div, a wonderful scientist. He won the Nobel Prize in the 70s, and he discovered this wonderful organelle that I told you, the lysosome. And as I told you, it was known that the lysosome degrades proteins. Not only that, but it was known also that the lysosome requires energy. Why it requires energy? Because the intralysosomal pH is about 5.5. And the cytosolic pH is around 7. So in order to pump log and a half of hydrogen inside, you need pumps. And indeed, the pumps were cloned, and we know about it. There are ATPases here sitting in the membrane. So protein degradation is there. At least people admit that proteins are degraded. They are not stable. Energy is required in the lysosome. Then what's the problem? Now, everything is solved. So what nevertheless attracted us to the field? What attracted us to the field was partially this table. So this is taken from a review of Fred Goldberg, who is a good friend of us at Harvard Medical School, and he just compiled data. And he showed that different proteins have different half-lifetimes. As I told you, some proteins turn over 20 times an hour or 10 times an hour, and some proteins live 2 days and 10 days. So how can it be? Because if you look at the lysosome and the mechanism of the lysosomal action, you see, that's what I told you. It's pinching off. You see this pinching off. So you pinch off droplets of the cytosol, you make vacuoles, and then the vacuoles open up and pour their contents into the lumen of the lysosome to be degraded. That cannot be selective, because in this droplet, all the proteins are included. It's like if I take a nylon bag and go and sample air in this room, so if I sample it in this corner or I sample it here, it will be the same air. 20% oxygen, 21%, 80% nitrogen, and the rest whatever. So maybe if I take it next to your nose, it will be a little bit more carbon dioxide when you expire air. But it will be the same. So all the proteins are here. So short-lived, long-lived, intermediate-lived. So how can it be that some proteins are living so shortly and some proteins are living forever? This cannot be explained, because once you are getting exposed to the mixture of proteases you are dead anyway. So there is no way out. Not only that, but how could it explain cell cycle? Remember the word cycling that was coined by Tim Hunt, who won the Nobel Prize among the three for discovering the cell cycle. Cycling is cycling. How it's cycling? It's being synthesized, get to mitosis, bang, it goes away. Again, up, down, up, down. So this can never explain it. How does the lysosome know to collect all the cycling from the cell? The lysosome is an organelle. It cannot go and swim around and 5 minutes before mitosis start to call cycling to come in and keep the rest. It doesn't work like that. So you can imagine. So the energy requirement, which was partially explained by the lysosome, but nevertheless, and the specificity and the cell cycle told us something is missing. We need to look for something else. So we started with the reticulocyte. We started with the reticulocyte because the reticulocyte was a good example of a cell that degrades quickly all its mechanisms and all its organelles, but doesn't have a lysosome, because the lysosomes are expelled already at the very initial stage of differentiation. So there are no lysosomes, but nevertheless degradation goes on. We were very lucky to start with it, because even after several years of working on the reticulocyte and coming out with all the findings that we had, people told us, yes, but reticulocyte is a terminally differentiating cell. So if whatever you discovered is important or is true at all, it's a niche in biology. It's not belonging to biology. You are working on a terminal differentiating cell, which gave us another 5 years to work quietly without competition. Because if you would have started with a nucleated cell, the whole world would have been after us. So, from time to time, we didn't plan it like that, I must admit. But it's easy. I remember people told me in conferences, oh, reticulocyte, yeah, you found something that is, unique to one single cell in biology. And it's maybe nice, but who cares about it? So we started with the reticulocyte, and only at the beginning, we published, I always show this, some of you saw it when, we published the first paper in BBRC in '78. This was the first paper in the history of the ubiquitin system. And it's today people don't look at this journal. It's so-called low impact factor. At the time that we started to work, the word impact factor was not in the vocabulary because there were no computers and no PubMed and no nothing. So who knew what is an impact factor? But now everybody knows what's impact factor. This paper is one of the two papers that was cited by the Nobel Committee as the founding important paper, which tells you that doesn't matter where you publish, but it matters what you publish. In my opinion, it's the most important paper I've ever published. And I want to point out to 2 points, one humoristic and one why it's important. The humoristic one is the following. I started my PhD with Avram Hershko, and 2 months into my PhD, he left. He went to a sabbatical, left me alone, and I did all the work here I did by myself. And I sent him the data, obviously no email, no nothing. I sent it by regular snail mail. And he wrote the paper, and he published it. And it was okay. 20 years later, I'm up to my last promotion to become full professor in my university, and a professor of pharmacology. And obviously, when you are promoted, like here, probably you have to write why you think you deserve the promotion. So I wrote a lengthy essay why I think I deserve it. And a professor came to my room, the professor of pharmacology, and said, Aaron, there is something wrong about your writing. Because I wanted to do analysis again. I'm talking impact factor and bibliometrics. And this paper that you cite as your most important paper is not in PubMed. Cannot find it. I said, what do you mean? So BBRC is in PubMed, everything. So indeed, I went into PubMed. It's not there. I went under my name. It's not there. And I looked and looked and looked, and I couldn't say anything. I said, what do you think, that somebody fabricated it, or there was another Aaron in the lab that did it, or what? And then I quote the issue, that Avram misspelled my name twice. My name is spelled twice. First of all, he put an H here, which is okay because it's the first name. Typically in English, you write double A, A-A-R-O-N, but that's okay. Then, in the family name, he missed the C here. It's C-I-E-C-H-A, not C-I-E-H. It's the name of a Polish city. I have nothing to do with it. My father came from the city of Ciechanover in Poland. So it's the name of a Polish city. So Avram didn't know. 2 months into my PhD, he went away. And then he wrote the paper. At that time, he even didn't bother to send it to me for proofing because it was from America to Israel 2 weeks and then back 2 weeks. So it was a long story. So he published it. Obviously, this paper is not on my PubMed. No wonder it's not on my PubMed. PubMed doesn't know me. PubMed knows only spelling. So this author has a single paper on a CV. But it just shows you, we are joking, but it's a culture that we invented, we foster, we nurture, and we grow up, and then we succumb as victims to the same monster that we built. So now, why the paper is important? Because the paper was a paradigm shift. And I talked to the students today about paradigm shift. So we got what we wanted. We took high-speed supernatant at neutral pH. Actually, it's a little bit above neutral. We did it at pH 8. And we got ATP-dependent degradation. So no lysozyme. And it cannot be even lysozyme because we are working at pH 8. Lysosomal enzymes don't work at that pH. And we got nice activity. Without ATP, with ATP, 7-fold stimulation. We used tritiated globin as a substrate. It doesn't matter, we used many substrates. Then, obviously, biochemists like to see pure protein. And there is a big difference today between today and those days about how you purify protein. Today, you purify protein by cloning, and by putting a tag and by pulling down. That's the way to purify protein, right? Easy, very easy. At those days, you started with 5 kilo of tissue, and you went to the cold room for 1 month, and you ended up with 10 microgram of a protein. That's how it works. These days, no cloning, no molecular biology, no techniques, no nothing. But so we wanted to isolate this ATP, elusive ATP-dependent protease, and typically we were captured with a notion that the protease always have a tango of two. So there is a protease and a substrate, like trypsin and chymotrypsin. It's trypsin. If you take a substrate, you give trypsin, it will be degraded because most proteins will have lysine or arginine in it, and trypsin cuts next to the C-terminus of basic amino acid. Chymotrypsin, aromatic amino acid, tyrosine, phenylalanine. So we thought, okay, we'll purify, and we'll end up with a protease that somehow acquired the unique property of being an ATP-dependent protease. Here we put the lysate, the crude lysate, on a column, on an ion exchange column, and some of the proteins came through the column, some of the protein adsorbed to the column, and we eluted them with high salt. Probably people don't do it anymore, but that's biochemistry, classical biochemistry. The protein that came out didn't have any activity. The proteins that were eluted with high salt had little activity, and we had to put the two together, 1 and 2. Now, this is a paradigm shift, a major paradigm shift, because from a tango of 2, we move to a tango of 3. Well, we can dance 3, it's okay, but this is not biology. We have 2 fractions that are needed to degrade the protein and the substrate, so this is 3. This is a paradigm shift. Why? Because remember, fraction 1 and fraction 2 are crude fractions that are split by virtue of the characteristics of the protein behavior on the column. So fraction 1 has 10,000 proteins, and fraction 2 has 10,000 proteins, more or less, doesn't matter, 5,000, 500. So nobody told us that fraction 1 has only 1 active protein in it. It can be that it has 1A and 1B, and if we'll take fraction 1 and put it on another column, it will split into 1A and 1B, and 1A and 1B both will be required, along with 2, in order to degrade the protein. So that's indeed what happened. So without taking you too long, warm raincoat, winter coat, cold room, and columns, 1 by 1 by 1 by 1. It took me about 5 years, and when I left the lab to MIT, I left behind 14 proteins, 14 proteins, that were all needed to be present at the same time in the same tube in order to degrade the protein. Now 14, what does it mean 14? We call them 1, 2, 3, 4, 5 until 14, but we knew their function. I don't have the time to tell you how it was like a puzzle that we put together. Who is first? Who does the first? So protein 8 started, and then it moved to protein 1, and it was all numbers that we gave to the protein, but that doesn't mean that this is the order of action in the reaction cascade. Think about the Krebs cycle. Think about glycolysis. There is an order. You take glucose, you phosphorylate it, then you phosphorylate it the second time, then you cleave it to 2, 3 carbon compounds, and then each of them goes into the Krebs cycle. There is an order, and here too there was an order. We found the order and we found the function. So we knew it took us 5 good years to do it with some help of Ernie Rose and others. I will not go into it. But this was just the tip of the iceberg, because the real story came out not by us, but in 2000 when the human genome was unraveled. And it turns out that the system that we discovered has close to 2,000 components. 2,000. It's almost 10% of the human genome. 10% of the human genome, actually a little bit less. The human genome, to be accurate, has about 22,000 proteins. The number of components of the ubiquitin system, we still don't know the finite number because it's coming up all the time, is about 1,800. So 7-8% of the human genome is the ubiquitin system. So it's huge. We just discovered the very beginning, and we were extremely lucky to do it, because we could have lost, I don't want even to tell you, we could have lost the way at every corner of turning. When we think about it, but we need luck too, and therefore it was a wonderful story of reconstruction. So let me tell you how the system looks now, and what is the story, and then tell you why the system was evolved at all. Why did we need it like that? It's because it's an awfully cumbersome system. Okay, So we have a protein that needs to be degraded. It doesn't matter whether it's faulty, denatured, inactivated, mutated, not necessary anymore. Proteins typically will not be degraded constitutively by the system. Something must happen to them. As I said, either oxidation or something. Once something happens to them, phosphorylation, they are being recognized by an enzyme called ubiquitin ligase, which we called at that time E3. Why E3? Again, the numbers. E1, E2, E3. This goes back to the history. Meanwhile, we realized that these people cannot communicate like that, so we gave them real names that reflect the function. So the E3, initially we called it E3, but then we called it ubiquitin ligase because we know that it ligates ubiquitin. The substrate binds to ubiquitin ligase. There are about 800 of those. So out of the 1,800 components of the system, about half are ubiquitin ligases, and they carry the substrate specificity of the system. They are the specific recognition element. So some will recognize some protein, some will recognize other, and other, and other. So this protein, something happens to it. In this case, it's doubly phosphorylated. I like this protein. Doubly phosphorylated, it's I-kappa B alpha. It's the inhibitor of NF-kappa B. We have been working on NF-kappa B for a long while, so I brought it as an example, but it doesn't matter. Something happens to it and it binds. So it's first handcuffed. It's scaffolded to an altar, if you think in a church terms. Then in parallel, a small protein, which is called ubiquitin, it's a highly conserved, evolutionarily conserved protein, is activated by ATP. We know the exact mechanism of activation. We worked it at the molecular level. By an enzyme called E1, there is a single E1 in the database. From E1, it's going to E2. And from E2, it's going to the substrate. And it's being conjugated covalently. It's really a peptide bond. So the first ubiquitin goes to the substrate, the second to the first, the third to the second, and so on and so forth. So they bind to one another to build what we called a trans-recognition proteolytic signal. So it's a recognition element in trans. Why? Because there is a trans element to recognize it. Now I don't have time to go into the chemistry, but the chemistry is rather complex here because the linkage is between the C-terminus of one ubiquitin, which is glycine residue, to an epsilon amino group of a lysine residue in the previous ubiquitin. But there are 7 lysines in each ubiquitin. Typically for degradation, it goes to one lysine, which is lysine 48, but it can go to any of the other lysines, but then it will not go to proteolysis. It will go to other functions. So gradually, just to tell you now, remember, we didn't mean to discover the ubiquitin system. We didn't know what ubiquitin is. It was discovered during our studies of a biological question. We started from a biological question. The system came later. It's the system that we looked for, but we didn't know what it is. Meanwhile, many functions were discovered to ubiquitin conjugation, in signal transduction, in carrying protein to their subcellular destinations, and so on. And all those require different chains where the ubiquitin, the second one or the third, is binding to either lysine A or lysine B or lysine C or lysine D, E, F, and G. So each binding has a different meaning. There is a whole code for reading the chain. In this case, this ubiquitin is bound to this ubiquitin. The C-terminus of this ubiquitin, which is glycine, is bound to this ubiquitin, to lysine 48 of this ubiquitin, and so on and so forth. And this ubiquitin binds by its C-terminus glycine to lysine 48 of this, and so on and so forth. So there is a whole code of ubiquitin. Unlike phosphorylation, ubiquitination has a code, which is much more complex. I will not go into it. So now we generated the signal. The signal is being recognized by a 2 million Dalton protease. It's a huge protease. It is made of 2 complexes, a 20S complex here in the middle, a 90S complex, which is a recognition complex. So ubiquitin binds here. It brings the substrate closer. And once the substrate is coming closer, there are ATPases here that open up the substrate and destroys it. To peptides, not to amino acids. And that's very important because already here we see one of the million functions of the ubiquitin system, because these peptides are being recognized by MHC class I molecules of the immune system. So if the proteins are derived from endogenous cellular proteins, nothing happens. But if this is a viral protein, the yellow one, there will be peptides that are derived from the virus or any other. It will be recognized by cytotoxic T cells, and they will kill the presenting cell. So the ubiquitin system, one of the many functions of the ubiquitin system, it serves as the proteolytic arm of the immune system to defend the body from foreign invaders, like viruses, because it will kill the presenting cell. So this is where the virus is replicating, obviously. So this is just one of the functions. Okay. So this is the ubiquitin system. So before I jump to the second part of my talk, let me just tell you in 2 words why we needed that. We needed it exactly for what I told you, for specificity. The lysosome, with its engulfment mechanism, couldn't pull out one protein of the same species out of 999,000 and leave all the rest alive. Same, the lysosome couldn't pull out one group of proteins like cyclin A or cyclin B and take it out of all the rest of the proteins in the cell and leave all the rest alive. For that, you needed exactly the ubiquitin system. You needed the cryptic protease. Remember, the proteasome, the protease is cryptic. It's not trypsin that floats around and destroys everything. It can float around like a shark that will not touch anything unless you are ubiquitinated. So ubiquitination is the secret here, the signaling. The protease is a naive, innocent, good boy, puppet protease. Nothing. It will not touch anything unless it's ubiquitinated. It's like a shark. A shark can swim next to you, will not touch you unless you bleed. So the shark senses something, exactly here. So once you have a deformed molecule, let's say an inactivated molecule that is misfolded or oxidized or whatever, the ubiquitin system will jump on it, ubiquitinate it, and only then the protease, the cryptic protease, the secret is signaling that lives side by side and acts together with a cryptic protease. You need signaling on one hand and a silent protease that will take only the signal proteins. This is the secret of specificity. The lysosome could never do it. Unless the lysosome would have evolved 10,000 different receptors, like ligases, but that sits on the membrane and pull out proteins and then transfer them to the lysosome. So you can imagine something like that, but it will be less efficient because the lysosome is an organelle. So everything stands still. How the lysosome reached the cell membrane or anywhere, while the proteasome and the ubiquitin system are floating around and can reach any corner in the cell. So even if we would have imagined a parallel mechanism of specific receptors on the membrane of the lysosome, it could have never been that efficient and fast, like a soluble cryptic protease side by side with the signaling mechanism. So now we understand also the evolutionary need in such a cumbersome system. And again, it's specificity. But it needs to be cryptic protease because an open protease will be a murderer, and then a signaling mechanism so the protease will recognize the signal molecule and pull it out. So that's the evolution of the system as we understand. Okay, let's go quickly to medicine. So why all the system is important in medicine? Why there are diseases. Now, I can tell you, now we can write a huge textbook on diseases that related to the ubiquitin system, but we need to understand why is it. So normally proteins are in a steady state, and pathologically 2 things can happen to them. Either they will be excessively degraded and go below the steady state, or there will be a mutation in the ubiquitin ligase or the recognition site of the protein. Let's say that the phosphorylating a serine residue on I-kappa B will be mutated to alanine. Then I-kappa B will never be, the inhibitor of NF-kappa B will never be degraded, will keep on inhibiting NF-kappa B, and we shall never get activation of NF-kappa B. So one mutation on a serine residue on I-kappa B and finish the inflammatory response. That's it. You can wipe it out because I-kappa B is an inhibitor of NF-kappa B, right? So I will not go into NF-kappa B, but I just give you an example how you can abolish a whole process by a single serine to alanine mutation. So protein will be accumulated. Now, in any of the reasons of why we degrade a protein, I can give you not a lecture, but a whole course of diseases that are related to it. Either it's quality control, or control of processes, or differentiation morphogenesis. So think about the brain. All the neurodegenerative diseases, with almost no exception, from Alzheimer to Parkinson to polyQ, Huntington disease, are diseases in which we see accumulation of aggregated proteins in the brain. Those aggregated proteins should be degraded, but they are not. Again, it's not the default of the ubiquitin system from time to time. But almost with no exception, we see accumulation of aggregated proteins in the brain. And what we see here, these are sections of brain which are immunohistochemically stained for ubiquitinated proteins. So the proteins are ubiquitinated, but the proteasome cannot take them. It's constipated. In this case, these brains are early onset Alzheimer patients. I don't want to go into the genetic defect here. Most cases of Alzheimer are sporadic and late age. These are young age and genetically related. I will not go into it, but you see here that with no exception, the brain is overly loaded with aggregated ubiquitinated proteins. So the antibody that stained these brown flames is an anti-ubiquitin antibody. Okay, let's go to cancer. Cancer is an interesting case because most of the drugs that we have today are anti-cancer drugs. So we can think of two cases. If a tumor suppressor like p53 will be excessively degraded, we shall have cancer. Or if an oncogenic protein, I talked to you about Wnt signaling, like beta-catenin. If beta-catenin will be not degraded, beta-catenin will signal to the nucleus via its transcriptional activity, and there will be activation of malignant transformation along with other factors. So it depends. Either a growth-promoting protein, not an oncogenic, a growth-promoting protein that will be under-degraded, or a tumor suppressor that will be excessively degraded, we shall have a problem. Here, human cervical uterine carcinoma, which is caused by the human papillomavirus. The human papillomavirus, this is another Nobel Prize to Harald Zurhausen from Heidelberg in Germany. The human papillomavirus encodes an oncogene that is called E6. E6 heterodimerizes to p53. The ubiquitin system looks at this heterodimer, sees something strange, and flushes p53 down the sink. And that's exactly what the virus is looking for. It's looking for removal of a tumor suppressor, because it's not a tumor suppressor, actually it's a quality control checkpoint protein, p53, because it's a small DNA virus, the HPV, and the HPV, in order to replicate and propagate, has to cut the DNA and to integrate its small genome into the cellular genome. The p53 would have sensed this integration event, the cutting and the integration, and would have stopped cell cycle and attempt to repair the damage or the integration, and if not, induce apoptosis. So the virus evolved the mechanism to shut down the emergency system of the cell before the integration event. And this is an early gene. So it's like an SV40. You have the early genes that are important for the infectivity, and only then you activate the mechanism, the hijacking mechanism. So this is exactly the case. And here, there are many examples. I just brought beta-catenin and the APC, that is the bed on which beta-catenin is degraded, the adenomatosis polyposis coli. It's not the APC of the ubiquitin system. There is another APC. It's the adenomatosis polyposis coli. So if beta-catenin, again, will be mutated in one of the serine residues that normally targeted for degradation by the beta-TRCP ligase, beta-catenin will be active, will be stable, will accumulate, will transcribe. This is the work of Hans Clever's and Paul Poliakis and many others, and we shall start to induce malignant transformation. And the same is for the EGF receptor. I can bring you many examples. So you see how either excessive degradation or inhibited degradation can lead to malignant transformation. So let's go to drugging the company, the system. You may think of, if you are developing drug, you may think of the best place to drug the system is here, where the ubiquitin ligase recognize specifically the substance. Because here, you expect to be at the highest specificity and the least side effects, because you are at the broadest point of the system. But it turns out that this was very difficult. It's only now, after almost 20 years that the system has been known, that people start to to drug this point. The first drug, the very successful drug, didn't target this point, but rather an unexpected point, and that's the proteasome. If you think of it, the ubiquitin system has a diamond shape. It tapers at the top with a single E1 that activates ubiquitin, go to about 50 E2s, to about 800 E3s, and then to about 10,000 substrates. And then once the substrates are ubiquitinated, they all are being funneled to the proteasome. So the system has like a diamond shape. So the first inhibitor, which has been in use for more than 10 years, extremely successful, with more than $5 billion sales. We have nothing out of it. We're stupid enough not to patent anything, just to publish, which is okay. I don't really regret it. Is a proteasome inhibitor. And you may wonder, how is it that you can inhibit the proteasome and nevertheless survive? I mean, if the ubiquitin system is so important, how is it that you block the protease that degrades every single ubiquitinated protein and nevertheless survive it? I'll try to explain it in a minute. So it's an inhibitor to the proteasome, actually to the active sites, which sit here in the middle of the 20S complex. And it's called, this is the first generation, but there is now a second and a third generation. This is bortezomib or Velcade, and it's being used in multiple myeloma, which is a B-cell leukemia. It's one type of leukemia. It's very common. Among the leukemias, the blood cancer, it's more than 20%. It's 20 to 25% of all blood malignancies. And here you can see a patient. It's a B-cell leukemia. So you remember, B-cells are the cells that secrete immunoglobulins. So you can use the immunoglobulin in the serum as a biomarker. And you can see here the galloping level in the patient. Chemotherapy started, but the patient relapsed. The disease started again. And here you see treatment with the proteasome inhibitor, and you go to nothing. So the level of the tumor goes down. And you can see here nicely the bone marrow of the patient. And you can see here the malignant bone marrow, with more than 40% of the cells are malignant. And following treatment, the malignant cells go to less than 1%, and the bone marrow is revived. The normal precursors of red blood cells and white blood cells are coming back. And here you can see another disease. It's a non-Hodgkin lymphoma. It's a parallel of multiple myeloma. And you can see a CAT scan. You can see the tumor sits here in the hilum of the right lung. You can see here, this is the sternum. This is the vertebra, the spinal canal. This is the air in the right lung, the left lung, the big vessels. And following treatment, you can see here that the tumor regressed significantly. What happens here? It's eliciting of the unfolded protein response. So the tumor making immunoglobulins, and the immunoglobulins out of, let's say, 100 molecules, one is misfolded. So normally the ubiquitin system will take it. But if you inhibit the ubiquitin system, not completely, only partially, and these abnormal immunoglobulins accumulate, and what they induce the unfolded protein response that was discovered by Peter Walter in UCSF, and then the cell apoptose. So it's not a chemotherapy. The drug has much less side effect than typical chemotherapy. It's an inducer of an unfolded protein response and apoptosis. So that's the beauty of it. So again, we don't inhibit the ubiquitin system completely. We inhibited it only partially. And because the cell is a factory to make antibodies, part of the antibodies are abnormal because they are misfolded inside the cell. And normally the cell will handle them. But in this case, when the ubiquitin system is partially inhibited, then these proteins will accumulate and induce the UPR. Now, let me take you, I don't have time, I want to take you to the last way, because there are many drugs that are, think about the ubiquitin system. You can inhibit the E1, the E2, the E3s, the proteasome. You can inhibit the recycling enzymes. There are enzymes that recycle ubiquitin back to the system. But I really want to take you to recent development and to show you the breakthrough that occurred in recent months. And that's Protax, molecular glues. It came by serendipity. It came by serendipity, by a serendipitous discovery that preceded understanding of the mechanism. I don't know, most of the faces that are seen here are young, but some of you, the old ones like me, remember a drug that unfortunately was allowed to use only in Europe and not in the United States. It's called thalidomide. Anybody knows about thalidomide? You remember thalidomide. Okay. Thalidomide was developed in Germany in the mid-60s as a drug to avoid vomiting during pregnancy, for women that vomit during pregnancy. And it was given to the woman. Many women took it. And all of a sudden, people realized that it was very quickly that the children that are being born are completely deformed. Many of them. They have eye in the middle of the forehead, one limb, fused fingers. I mean, horrendous teratology, horrendous. The linkage to thalidomide was made immediately. The drug was forbidden. How America was saved, it was a disaster. I think that more than 2,000 kids were born in Europe until people started, in Holland, in Germany, in France. And people realized it very quickly, and it stopped. The Americans suspected that the Europeans, the licensing of the drug went through really rigorous procedures, and the FDA didn't let it go into America, so it was forbidden from sales in America. And thalidomide was forbidden, and it was forgotten. Actually, when the word thalidomide was mentioned, people thought, wow, this is monstrous. So they even were afraid to approach it from a distance. They thought that even the vapors may kill them. But though the drug is not obviously vaporizable, so it doesn't matter. And all of a sudden, people discovered a few years ago that thalidomide kills B cells. And nobody knew how. And only 2 years ago, the mechanism was discovered. Meanwhile, thalidomide was entering the market as a drug for multiple myeloma patients, initially by license, so each patient had to be reported because, again, people were afraid that it will evaporate in the middle of the streets of New York, and pregnant women that walk to the Empire State Building will breathe the air and will get a teratogenic effect on the embryos. So it needed for every patient a license. Then it was released gradually. People understood that it's just... Anyway, it turned out that thalidomide is a molecular glue. Now, what is a molecular glue? Molecular glue known also as what we call PROTAC, or proteolysis targeting chimera. So what is a chimera? You have here a ligase, okay? E2 and E3, and you have here a substrate. And the substrate is not degraded. Let's say that you have a mutated MYC, okay? And we want to degrade it because it's oncogenic, or mutated P53, or mutated RAS. You want to degrade it, but you don't know the ligase and you don't know anything. What you do, you make a protein, a chimera, a small molecule that is made of two heads, a yellow head and a red head. Now, the yellow head binds to the ligase. So you find something that binds to the ligase. Okay? You can make a screen. You take a ligase and you make a screen of small molecules that bind to it. Then you made a separate screen, and you find another molecule that binds to the target. And then you fuse them. And then you put this molecule into the cell, and it will bring the two together. Once you bring the substrate and the ligase together, the ligase will ubiquitin at the substrate and kill it. So the ligase is very promiscuous. The ligase doesn't care which substrate it has. Normally, the ligase will bind only specific substrate. Wow. How? By being the substrate coming in the right vicinity to the ligase. But if you bring it artificially to the vicinity, then it will do it anyway. How do you do it? By small molecules that glue them together. It's called molecular glue. It's a chemical term. It turns out that thalidomide is a molecular glue. It's a natural molecular glue. How? Here is thalidomide. Actually, I can show you it here. Here is thalidomide. And thalidomide glues Aiolos or Icaros, which is a transcription factor that drives oncogenic B cells, oncogenesis in B cells. It brings it to the ligase, which is called cerebron, CRBN for degradation. Without the blue one, the red one will never bind. Actually, the scheme is not correct because the red one binds to the blue that binds it to the CRBN. Okay, here it's a little bit better. You can see it here, that this one, the thalidomide, brings this one to cerebron. But it's a small molecule that goes into the cell and bring two components together that otherwise will not recognize one another. So it's a glue. As I said today, if I can take a piece of wood and a good glue, I can stick it to the wall. The wood will never go to the wall unless I put it together. You need a glue for that. And the glue is completely artificial. And it turns out that thalidomide somehow glues together two transcription factors, Aiolos and Icaros, that drive the oncogenic process by driving MYC and by driving interleukins. And, those two transcription factors, Icaros and Aiolos, drive the whole oncogenic process of B cells. Now, if we degrade them, we stop the process. We just force them to be degraded. How? By gluing them to the ligase. So, and here you can see the same. And papers were published just a few months ago. So here you can see lenalidomide, which is a derivative of thalidomide, causes selective degradation of Icaros 1 and 3 in multiple myeloma cells. Here was a paper that came a month ago, or 2 months ago, in Nature. Lenalidomide induces ubiquitination degradation. In this case, it's a different one. It's a different glue of CK1 alpha, which is a kinase in myelodystrophic syndrome. Now, this opened a whole new platform. Actually, we have half of the platform. Think about it. We have half of thalidomide already ready. So we can take half of thalidomide that we know from the structure. The structure was worked out beautifully by Nicola Thoma in the Friedrich Mischer Institute in Basel. So we know exactly the structure. We know what in thalidomide binds to the ligase. So we can take half thalidomide and that bind to the ligase. So we have half of the story done. We can convert the ligase into universal ligase that will degrade anything. And then let's say that we want to degrade RAS or MYC or whatever. We now start a screen of molecules that bind in high affinity to RAS. Doesn't matter. We can screen, we shall find. Or MYC or P53. And then we make a chimera between the half that binds to the ligase and the half that binds to RAS. And this will lead to degradation of RAS. And we know already that we can do it. So that's a big promise for pancreatic cancer and for many other cancers. Obviously, not all chimera will not be toxic. Not all of them will be cell-penetrable. But it opens also we don't need to stick to thalidomide. Thalidomide is ready already because we know it. We can take any ligase in the world and screen for binders to the ligase and then screen for binders to and then we can mismatch. We can just do a mishmash. So once we shall have a molecule that binds to 10 ligases, then we can fit them according to solubility, to cell permeability, to halves that bind to the substrate. So we are now gradually increasing the library, and there is not a single company that is now out of the game. Merck, Johnson, Novartis, Sanofi, they're all in the game. Each has their own target, and some of them are working on the same targets. But I feel now that the field is breaking, people were very much disappointed with the Ubiquitin system, the drug companies, because they said, look, the system has been there for more than 15 years, and there are no, except for the proteasome inhibitors, there are no drugs. Well, we understand why now, because if you think about most of the drugs that are being used in cancer are kinase inhibitors, many kinase inhibitors. But to develop a kinase inhibitor was relatively easy because most of them are sitting on the ATP pocket. So now the structure is known, so it's easy relatively to develop kinase inhibitors. Here, the ubiquitin system, it's not small molecule, it's the substrate that interacts with the ligase. So it's a protein-protein interaction on large surfaces and low affinity. So it was very difficult to develop. Many companies tried very hard and failed until this came out. And now, I think that it will break open the field. Think about degradation of aggregated proteins in the brain. We shall make screening for polyQ proteins, for Huntington, for spinocerebellar ataxia, for Alzheimer, for Parkinson, and so on and so forth. So the field is really open, and I believe that in 5 years there will be blockbuster drugs to many diseases that are now not approachable. So it just takes time. So let me finish by just thanking some of the people. I didn't show you any specific work, but I started as a grad student with Avram, and then Ernie Rose, we had to go to Philadelphia because it was complicated protein chemistry, all this conjugation. We didn't understand anything there. And with the help of Ernie, and we kind of able to pave our road into understanding the conjugation mechanism. And then I collaborated when I was at MIT with Alex Warshawski and Dan Finley on the first nucleated cells that degrade, that have a mutation in the ubiquitin system. So that took the system from the strange, awkward reticulocyte into the world of biology, because we discovered this first mutant, nucleated mutant that is defective in the system, and then obviously many postdocs and graduate students. Let me just say one word of summary before I conclude. We started, as I told you, I tried to tell you in a marathon run, with a question that had assumptions, that there may be something energy dependent and so on. So first of all, we started with a curious biological question. We had no idea about the ubiquitin system. We didn't know anything. We just knew that something is missing out of some milestones, and we decided to go and find it. It was risky, but people take risks, and it was discovered. But I think that more than that, which is important, and I face it a lot in my own university, I'm sure that you face it here, universities and governments and funding agencies are pushing today to what we call translational research or applied research, which I think is the vastly wrong approach. Because if you do translational research, it's good to do translational research. Obviously, we need to develop drugs, and we need to, but if you are being pushed to it and do only that, at the end you will not discover anything. The drugs to the ubiquitin system, the proteasome inhibitor, could have never been discovered without discovering first the proteasome. How can you discover a drug to a target if you don't have the target? Thalidomide is a different story. It was serendipity. But mostly we don't discover drugs by serendipity. Not nowadays when we are developing personalized medicine and understanding mutations and so on. So in the word applied or the word translational, we translate something. We translate basic to practical. We apply basic to practical. If there will be no basic, there will be no translation because there will be nothing to translate. So I think that there is a dangerous trend here that people don't realize, governments and politicians don't realize what is the danger of drying out basic research. We started from curiosity, and all of a sudden we find ourselves in the highway of drug discovery. So I think that this is the way to go. So for me, when I am asked about translational medicine or translational research, I say that I don't know what it is. I know only what is the difference between good science and bad science, but I don't know what is translation, because from good science, good things are coming out. So thank you very much.
Well, thank you very much for a beautiful talk. I think it was a great summary of how the discovery is made, also for many of the students, the ideal presentation, so how the science is being done. And without first giving the time for questions, I would first give Aaron the MENDEL Medal for the achievement in the science. So please give me his applause. And we have time for questions. So I will start. So you maybe touched a little bit about the complexity of the code. Not only that ubiquitin is conjugated to the same lysine, but the numerous lysines that are possible. Also numerous ubiquitin-like molecules, SUMO and NAT. So where do you think the whole system will stop? Because it seems to be more complex, so each different modification means a new enzyme which will be able to recognize this kind of modification.
Well, indeed, it's a very complex code, unlike phosphorylation, that we know that the SH, the downstream recognition elements in phosphorylation, here we still don't know the downstream. What we are truly missing is structure, and it's very difficult to do the structure of, you need to synthesize a chain in order to pull down the downstream interactors. And so first we need to pull them down, and then we need to understand the structure in order to understand what ubiquitin is doing there. So we are still not there as far as structure is concerned, but it will come. There is no doubt it will come. But decoding the ubiquitin code will take time. It will take time. We are not there yet.
Thank you for an excellent lecture. What do you think about proteins which can survive in 37 degrees years, maybe decades, in human, like cohesins, for example?
Yeah. Either they don't change, the ubiquitin system don't recognize them. What people claim is even cohesin, they don't live forever. What they do, they are degraded by the lysosome because membrane turn over, everything is turning over. I think that the default is the lysosome. So if you live long enough, at the end you end up in the lysosome because the lysosome pinches off. If people calculate it right, so for the lysosome to pinch off the entire cytosol takes about 40 hours. So by 40 hours, this little droplet accumulates to the whole cytosol. So in 40 hours, everything is gone, except if you are excluded. Let's say some cytoskeletal protein cannot be engulfed, like actin and myosin. So the default is the lysosome. But if you want to really rapidly remove a protein because it's inactivated, misfolded, and so on, then the ubiquitin system. So the ubiquitin system is the specific one. And then below the specific sits the default that is active kind of all the time. So that's I would say the task division between the two systems. One is active basically all the time, and the other one is recruited when necessary, to action.
Yeah. I have two questions, if I can. So the first one is that known what is the mechanism of the teratogenic effect of thalidomide?
The mechanism
of teratogenic effect of thalidomide.
It was discovered. It drives to degradation a transcription factor that is important for development, which is called MACE2. It's again the work of Nikola Toma. It's beautiful work. So it takes an extremely critically important transcription factor and trashes it. I think that it's MEIS2, MACE2 or MACE1, I don't remember. But this is an extremely important transcription factor. And it sheds it or it just flushes it down the sink.
The other question is there any similar, are there any similar molecules like thalidomide produced, or the molecular glues produced naturally by the human body or the pathogens?
I don't know of. But what people are doing today, they are making new derivatives. And actually they are screening, as I said, for new derivatives that will target their own proteins. So two derivatives are already on the market, besides thalidomide, which were mentioned. It's pomalidomide and lenalidomide. And they are degrading CK1 alpha and other proteins. But new ones will come soon. But they will all be synthetic.
Maybe I'll have one more question. So what do you think about, aneuploidy, which is associated with the cancer, so having extra copies of chromosomes, then shifting the balance of the protein content in the cell, and then how this perhaps, so rather than, so not
I don't think
having
that this
a
is
particular...
related to the ubiquitin system. The ubiquitin system, because this involves many proteins. And I think that the ubiquitin system is like, direct to the point. I think that this is more gross pathology because it will involve several ligases, and that my gut feeling is I don't know that the ubiquitin system doesn't play here a role unless APC. There is another APC which is the anaphase promoting complex. There is a whole confusion in the terminology between the APC of beta-catenin and the APC of the ubiquitin system, which is the ubiquitin ligase that is involved in degradation of many cell cycle regulators. But again, it has different components that endow the ligase with a specificity. I don't know. I mean, it's too complicated.
You mentioned in the beginning that 8% of the protein gets degraded every day. Do you know how much of this is via ubiquitin system?
Yeah, it's a good question because I didn't mean to mislead you to say that all is ubiquitin. Obviously, many extracellular proteins, it's about half-half. So extracellular proteins like albumin is also degraded. Immunoglobulins, circulating proteins, it's mostly circulating proteins, will be degraded by the lysosome via pinocytosis, asialoglycoprotein receptor, and the rest, so the estimation is not half-half, it's about 60% ubiquitin and about 40% lysosome. But the lysosome, again, it depends what do you think of, I would say 60% proteasome and 40% lysosome, because in order for the lysosome to degrade a protein, you also need ubiquitination. So the EGF receptor, for example, the insulin receptor are ubiquitinated, and only then they are like Cybl. C-Cybl is the ubiquitin ligase of the EGF receptor, though the EGF receptor at the end, because it's a membrane protein, will end up in the lysosome. So people are confused about it because ubiquitination has many more functions in targeting than the proteasome. The proteasome will take only part of the ubiquitinated proteins, but the lysosome can take the other part. In autophagy, the same. So we autophagocytose. So if you ask how many proteins the degradation is dependent on ubiquitination, it's 100%. How many proteins are dependent on the proteasome for degradation, it will be 60%. So, the system, it's a different view of the system.
Yeah, thank you for a wonderful talk. The question is, you mentioned quite several times during your talk the substrates for these pathways are very often transcription factors from the nucleus or other nuclear proteins. Yet proteasome is not localized in the nucleus. So how this is supposed to be a very fast system. So is it complemented by some transport system?
Yeah, it's a very fast system, but if I understood you correctly, I'm not sure that I understood you. The system is in the nucleus, full-blown, so you don't have to go out of the p53 MIC. So all these proteins degraded in situ in the nucleus. The system is also sitting on the ER, on the outside lamella of the ER. So there is a whole mechanism that is called ERAD, or ER-associated degradation. So proteins that are misfolded are being retrotranslocated from the ER outside and being actually captured while exiting the membrane, ubiquitinated and pulled out by the proteasome and VSP94. So there is a whole mechanism of pulling out. But they are degraded co-transportationally while exiting the ER. So the system is basically in most of the organs, in the nucleus and in the ER. Yeah, it's extremely efficient system.
So just wondered, you talk about how complex the whole degradation system is. How is it conserved between various organisms, like let's say yeast?
Well, it's ubiquitous. It's the same complexity. Yeast has it, and it's all over the place, but it's only in eukaryotes. So prokaryotes don't have the signaling mechanism. Mycobacteria tuberculosis has a very remote mechanism with a protein that is not homologous to ubiquitin, but it has some ubiquitin-like domains, which is called PUP. PUP. So in Mycobacterium tuberculosis, there is a process called papillation. But bacteria like E. coli has proteases like ClipX and ClipP that are very similar to the proteasome. They are much more primitive. That also degrade misfolded proteins, but a much more primitive mechanism. So there is the upper signaling mechanism of ubiquitination is missing. So basically, the ubiquitin system as we know it starts with the eukaryote, not even in archaebacteria. So it's POMB yeast have it in full blown, and then up.
Okay, thanks.
It's ubiquitous in plants, everything. I mean, they all have it.
So you talked about human diseases. I guess there will be quite a high potential to use it also against infectious diseases. Are there any attempts to use the glue system for like viral or even parasitical infections?
The question is whether the ubiquitin system also fights...
No, whether you could use as a therapeutic the glue system to fight against infectious diseases, if you would develop a specific recognition.
Well, there are many transcription factors like NF-kappa B that because of infection will elicit an inflammatory response to fight the infection. So it's coming
No, I meant straight ahead to find specific molecules or specific surfaces that would recognize proteins specific for.
Oh no, I get it. No, I don't know.
Or,
no, I got it.
Yeah.
No, I don't know.
Because this
yeah, it makes a lot of sense. Yeah.
Okay, if there are no more questions, please join me to thank one more time for a beautiful lecture.